Imagine a world where the actual cells inside your body, well, they could be precisely reprogrammed whenever needed. Where these complex, really life-changing therapies that used to mean weeks in hospital or, huge manufacturing plants, maybe they could come from something as simple as a standard IV infusion.
It sounds like science fiction almost.
It really does. But that vision, it's becoming reality much faster than I think many people expected. And right at the heart of this is a pretty audacious idea, making sophisticated CAR T cells directly inside the patient, not in some distant lab.
A huge shift.
Welcome back to BioTales, everyone. Today we're diving into the, frankly, remarkable story of Capstan Therapeutics. This is a company built on a truly innovative concept, merging mRNA delivery, which we all heard so much about recently, with cutting-edge cell therapy. And it's all happening right there, inside the bloodstream.
Convergence is key.
Exactly. From their founding, late 2022, to being snapped up by AbbVie in 2025. That journey is just incredible. It's not just about the science. It's about pushing boundaries, and it gives us a real glimpse into the future of treating things like autoimmune diseases. So how did they actually pull this off?
Well, what's so striking about Capstan is how they entered the scene with such a clear, almost bold vision. Their whole identity is really about that convergence you mentioned.
Right.
Taking the precision of targeted cell therapy, like CAR-T, and combining it with the speed and adaptability of mRNA delivery. This wasn't just tweaking things. It was a fundamentally new blueprint for tackling some really tough medical problems right inside the patient.
And the speed, founding to acquisition in under 3 years, that's lightning fast in biotech.
It really is, and it screams potential, right? The fact they reached a multi-billion-dollar acquisition so quickly tells you how powerful the underlying technology was perceived to be and now increasingly proven to be.
Okay, so to really get this, we need to rewind a bit. Back to the University of Pennsylvania. Can you take us there? Doctors Jonathan Epstein and Haig Agajonian, both cardiologists, grappling with a big question.
Absolutely. You have to picture it. They were wrestling with this really fundamental challenge. Could you actually engineer T cells inside a patient's body, make them target and kill disease cells, but skip that whole incredibly complex, expensive, time-consuming process?
The standard CAR-T process.
Exactly. The leukapheresis, shipping the cells, modifying them in a GMP facility, expanding them for weeks, then giving them back.
Right, so they wanted to bypass all that.
Completely. It wasn't just about convenience. It was about a potential paradigm shift in how we deliver cell therapy.
And this quest, it didn't happen in isolation, right? They brought in some heavy hitters.
Oh, definitely. This attracted key collaborators, people like Dr. Carl June. A giant in CAR-T therapy.
The pioneer, really.
And Dr. Drew Weissman, whose work on mRNA, it changed the world with the COVID vaccines. That combination of expertise was, let's say, catalytic.
So what came out of that initial collaboration?
Their early work led to the creation of these things called targeted lipid nanoparticles, or TLNPs. Think of them as tiny, sophisticated envelopes.
Like the LNPs in the vaccine?
Similar, yes, but with a critical difference. These carry modified mRNA instructions, but crucially, they're coated with protein binders, like little Velcro patches designed to latch onto very specific types of cells.
Ah, the targeted part.
Precisely.
And they showed this worked. In animal models, I assume?
They did, and it was quite elegant. They used nanoparticles targeted to CD5, a marker on T cells, to deliver an mRNA encoding what's called fibroblast activation protein CAR. It's a receptor designed to recognize and target specific harmful fibroblast cells involved in fibrosis. They delivered this into circulating T cells in animal models. And here's the really cool part. The T cells only express this CAR for a few days, transiently.
Just temporary.
Right. But in that short window, these temporary cells effectively cleared out the pathogenic fibroblasts and actually reversed cardiac fibrosis in the model.
Wow. So in vivo generation, transient effect, and actual disease reversal.
Exactly. And all without viral vectors, without taking cells out of the body. It was a blueprint for a controlled, non-permanent gene therapy delivered directly. That really opened up possibilities.
That kind of proof of concept, in vivo gene delivery without viruses?
Yeah.
That must have turned heads. That's not something that stays in an academic lab for long.
You got it. The science was ready to leap. By late 2022, Capstan Therapeutics officially launched, and not quietly. They launched with an incredible $165 million in initial financing.
$165 million right out of the gate.
Right out of the gate. And look who backs them. The venture arms of huge pharma companies, Pfizer, Bayer, Novartis, Lilly, BMS, plus top-tier biotech VCs like RA Capital, a real who's who.
That level of funding for a brand new company, especially in biotech, is extraordinary. It signals massive confidence from people who know the space. What specifically triggered that flood of capital?
Well, the timing was perfect. Just before Capstan officially launched earlier in 2022, Doctors June, Weissman, and their colleagues published this landmark paper in Science. That paper was the definitive proof of concept. It showed you could actually generate functional CAR T cells inside a living animal just by injecting mRNA packaged in a targeted nanoparticle.
So not just theory anymore.
Not at all. They showed a single IV dose in mice created CAR T cells that reduced fibrosis, even improved heart function in a disease model. That paper created a huge buzz, and this powerhouse syndicate of investors came together really fast around that data.
Makes sense. With that kind of money and the core science validated, building the right leadership team must have been absolutely critical. Who did they bring in?
They needed seasoned leadership for sure. They recruited Dr. Laura Shaver as CEO. She's highly respected, known for leading Synthorx to that huge acquisition by Sanofi.
Right, a proven leader.
Definitely. Then Dr. Adrian Bott, who previously headed research at Kite Pharma, another major CAR T player, he came in as chief scientific officer. A really strong team.
And their mission was clear.
Crystal clear. Take this revolutionary in vivo CAR technology and get it to patients.
Okay, let's dig into that technology a bit more because it really is the core of the story and why it's such a departure. For listeners maybe less familiar, remind us why traditional CAR T, even though it's been revolutionary in cancer, is just so difficult.
Yeah, it's worth retapping because it highlights the problem Capstan aimed to solve. Traditional CAR T is a logistical behemoth. First, you collect the patient's T cells, that's leukapheresis.
Right.
Then those cells get shipped, often frozen, to a specialized manufacturing facility. There, they're engineered, usually with a virus, to express the CAR receptor.
The targeting part.
Exactly. Then these engineered cells have to be grown, expanded into billions over, several weeks.
A long time.
A very long time. And before the patient gets them back, they often need chemotherapy lymphodepletion to make space for the infused cells. Then the infusion, followed by careful monitoring for side effects, often in hospital.
And the cost.
Astronomical. We're talking $400,000, $500,000, sometimes more, just for the drug product itself, not counting hospital stays, other treatments. It's custom made for each patient. One batch, one patient. Doesn't scale easily.
Okay, so Capstan's in vivo approach completely flips that.
That's the goal. Instead of all those ex vivo steps, the collection, the shipping, the lab work, the viruses, the idea is to reprogram the T cells right inside the patient's body.
Maybe with just an injection.
Potentially something as simple as that.
Yep.
You eliminate the cell harvesting, the viral vectors, the weeks of lab expansion, maybe even the preconditioning chemo.
Which would mean
an off-the-shelf product, something that could be manufactured consistently, stored, shipped, and used when needed. Much faster, potentially far, far cheaper. And that opens the door to using this kind of powerful cell engineering for diseases beyond just certain cancers like autoimmune diseases.
the key piece of tech here is the lipid nanoparticle, the LNP. We heard about them with COVID vaccines, but standard LNPs aren't very picky, are they? They tend to end up in the liver.
That's a major issue with conventional LNPs, yes. If you just injected LNPs carrying CAR mRNA, a lot might go to the liver or other unintended places, not necessarily the T cells you want to program.
So how did Capstan solve that? This is where their secret sauce comes in.
Exactly. This is the targeted in TLNP. Capstan developed their proprietary cell seeker technology. Essentially, they decorate the outside of the LNP with a specific binder, like an antibody fragment or another protein.
A homing device.
A molecular homing device, precisely. This binder is chosen to recognize a specific marker on the surface of the target cell. For their lead programs, it's often CD8, found on cytotoxic T cells.
So the TLNP finds and sticks only to the T cells.
Preferentially, yes. It ensures the mRNA payload gets delivered right where it's needed, much more efficiently than a non-targeted LNP. It's about precision delivery. And I think Dr. Adrian Boss explained it real well. He described it like this. Our targeted LNPs bind only the cell type we care about, deliver the mRNA payload, and then disappear. The cells translate the mRNA transiently, express the CAR receptor for a few days, do their job, and then return to normal. It's like giving the immune system a temporary software upgrade.
A temporary software upgrade.
Yeah.
I like that. It's intuitive. And that temporary part, the transience, is really interesting. Traditional CAR-RTs aim for long-term persistence. Why is temporary expression actually a good thing here, especially for autoimmunity?
That's a really critical point, especially for non-cancer applications. Initially you might think temporary is a weakness In cancer, persistence can be vital.
Right, you want those cancer-killing cells sticking around.
Exactly. But for autoimmune diseases, where the immune system is mistakenly attacking the body's own tissues, long-term persistence of a powerful engineered cell might actually be undesirable, even risky.
Okay, why?
Well, first, safety. Because the effect is transient days to maybe a couple of weeks as the mRNA degrades the risk of long-term side effects like chronic toxicity or issues for permanent genetic changes is dramatically lower. If there are side effects, they should resolve as the CAR expression fades.
Makes sense. Less long-term risk.
And second, perhaps even more importantly for autoimmunity, you often don't want to permanently wipe out entire immune cell populations. Take Capstan's lead program, CPTX-2309. It targets CD19 found on B cells.
Okay.
The goal isn't permanent B cell destruction. It's a deep, rapid, but transient depletion of the B cells, especially the pathogenic memory B cells driving the disease. You clear those out.
And then what?
Then you allow the body's natural processes to take over. Healthy, naive B cells can repopulate the system. It's like hitting a reset button.
An immune reset.
Exactly. You break the cycle of autoimmunity without causing long-term broad immunosuppression. That's the hope.
So a deep cleanup followed by a healthy restart, rather than just continuously suppressing the immune system like many current drugs do. Plus, that transient nature means you could potentially redose if needed, right?
Yeah.
Or adjust the treatment intensity.
Precisely. It offers flexibility, tunability. You could potentially give another dose down the line if the disease flares up again. It allows for more controlled, on-demand therapy.
Okay, so with this powerful science and the initial funding, Capstan got to work refining the platform.
They did. The team really buckled down, optimizing the TLNPs. And by November 2024, they presented some really compelling preclinical data at the American College of Rheumatology meeting. That got a lot of attention.
What did they show?
They showed their targeted nanoparticles successfully engineered CD8 T cells in vivo in non-human primates. And critically, this led to profound B-cell depletion.
The intended effect.
Yes, but here's the kicker. They achieved this without any chemotherapy preconditioning.
No lymphodepletion needed. That's huge for CAR T-related therapies.
It's a massive potential advantage. Remember, traditional CAR T often requires chemo, which has its own toxicities. They showed deep B-cell depletion without it. And importantly, afterwards, the animal's B-cell compartments repopulated with naive, healthy cells.
That immune reset concept playing out.
Exactly. That data really solidified the potential in autoimmunity.
And investors clearly liked what they saw. Because in March 2024, they raised another huge round, right? A Series B.
They did. A $175 million Series B, led by RA Capital again, and joined by others like Forbion, J&J Innovation, Mubadala, big names.
And I remember Nandita Shangari from RA Capital calling the platform differentiated and highlighting the potential in the, quote, enormous autoimmune disease market. That's a strong endorsement, especially when biotech funding was getting tighter.
It was a huge vote of confidence. It really signaled that the smart money believed Capstan was onto something big, particularly for autoimmunity, which logically set the stage for the next big step.
Moving into humans.
Exactly.
And that news dropped in June 2025. Capstan initiated a phase 1 trial for their lead candidate, CPTX-2309.
A major milestone.
The study started in Australia, evaluating safety, PK, PD. But here's the really interesting part. They started in healthy volunteers. Now, for anyone familiar with cell therapy trials, especially CAR T, that sounds unusual. Why start in healthy people?
It is highly unconventional for this type of therapy, you're right. Usually phase 1 for something like CAR T involves very sick cancer patients, slow dose escalation, often chemo. It's all about managing potential toxicity in a vulnerable population.
So why the different approach here?
It's actually quite strategic, and it speaks to the perceived safety profile of their transient non-viral approach. Because they don't need the toxic chemo preconditioning, and because the cells aren't permanently altered, the risk is considered low enough to ethically test in healthy individuals.
And what's the advantage of doing that?
Speed and clarity. In healthy volunteers, you can escalate the dose much more quickly than in sick patients. And you get cleaner data on the pharmacodynamics, basically. How much drug leads to how much B-cell depletion and for how long without the noise of underlying disease or other medication.
So you figure out the dose response much faster.
Exactly. You de-risk the dosing much earlier. So when they do move into patients with autoimmune disease, probably in phase 2, they'll likely already have a good idea of a safe and potentially effective dose range. It could significantly speed up development overall.
That makes a lot of sense. Get the dosing right quickly and cleanly before you go to patients who really need the therapy. And their chief medical officer, Dr. Ramin Farzana Farr, really nailed the goal, didn't he?
Yeah, his quote summed it up well. We want to hit the reset button on the immune system by transiently expressing an anti-CD19 CAR in CD8 T cells. We can clear memory B cells and then allow naive B cells to grow back. This could lead to durable remissions without continuous therapy.
Durable remissions without continuous therapy. That's the holy grail for so many autoimmune diseases.
Absolutely. And while CPTX-2309 was the lead, they weren't putting all their eggs in one basket. They had CPTX-2506 targeting BCMA for plasma cell disorders, and we're exploring other targets too, like for fibrosis and maybe even cancer down the line. But autoimmunity was clearly the main focus.
Then, the big news.
Yeah.
Just weeks after announcing that phase 1 start, right? End of June 2025.
Boom. AbbVie announces they're acquiring Capstan Therapeutics. Up to $2.1 billion.
$2.1 billion. That's a massive validation for a company that young.
Monumental. It sent shockwaves, really. AbbVie, a major player in immunology, basically saying, we believe in this platform.
How did AbbVie frame it?
Their EVP, Dr. Rupal Thakkar, talked about it fitting their strategy to transform care by actually resetting the immune system, not just managing symptoms, a fundamental shift.
And Laura Shaver, Capstan's CEO.
She highlighted how the partnership would accelerate the development of in vivo CAR-T across multiple autoimmune diseases. It wasn't just an exit, it was seen as validation for the whole field of targeted mRNA delivery and in vivo cell engineering. A watershed moment, really.
That AbbVie deal really underlines the perceived value. But getting there, it couldn't have been easy. Groundbreaking science rarely is. What were some of the major hurdles, the skepticism they faced early on?
Oh, absolutely. You know, every big leap faces pushback. The whole idea of delivering CAR in vivo without a virus, that was met with quite a bit of, can they really do that? Skepticism initially.
Because viruses are so good at getting genes into cells.
They are nature's experts at it. So mimicking that efficiency with a synthetic nanoparticle, especially getting it into tricky cells like T cells, required immense painstaking optimization. We're talking about fiddling with the exact lipid mix, how to attach the targeting protein, ensuring the mRNA was stable. Lots of hard work in the lab.
Let's linger on that viral vector point. Why was getting away from them so important? What are the real drawbacks?
It's a big deal. Manufacturing viral vectors is one of the biggest bottlenecks for traditional cell therapy. It's incredibly expensive, tens of thousands of dollars per patient dose, and slow. It doesn't scale well for large patient populations.
Cost and complexity barrier.
Exactly. Plus, there's the safety aspect. Viruses often integrate their genetic material into the host cell's DNA. While the risk is low, there's a theoretical concern about insertional mutagenesis accidentally disrupting a critical gene.
And that makes redosing tricky too, right? The immune system might react to the virus itself.
Correct. You can develop immunity to the vector, making subsequent doses less effective or impossible.
So Capstan's non-viral mRNA LNP approach neatly sidesteps all that.
That's the beauty of it. No viral DNA integrating anywhere. That lowers the long-term safety concerns and potentially makes redosing feasible. And LNPs containing mRNA can be manufactured using standard pharmaceutical processes, much more like making a regular drug.
Scalable, off the shelf, potentially much cheaper.
That's the promise. The real core innovation, the aha, was figuring out how to make a targeted LNP that could efficiently get mRNA into T cells, which are notoriously difficult to transfect.
So getting stuff inside them is hard.
Very hard. So those moments, seeing the TLNPs actually homing to the CD8 T cells in primates, that must have been huge. Then measuring that deep B cell depletion without chemo, another massive win. And then realizing this whole platform could pivot towards autoimmunity, a huge area unmet need, largely untouched by cell therapy. That was probably the biggest strategic aha moment. Each success like that chipped away at the doubts and built momentum.
So now Capstan is part of AbbVie. The journey continues, but under a bigger umbrella. What are the big questions now looking ahead?
Oh, plenty of fascinating questions remain. The biggest one, probably. Will this transient CAR expression be enough for long-lasting remission in chronic autoimmune diseases like lupus or myasthenia gravis, or will patients need periodic redosing? That's what the clinical trials will tell us.
Right. Durability is key.
Definitely. Then there's the platform itself. Can these TLNPs be engineered to carry different payloads, maybe mRNA for cytokines, or even gene editing tools like CRISPR? And can they target other cell types effectively? Lots of potential beyond CARs for T cells.
And scaling up manufacturing, ensuring consistent quality and safety for potentially millions of patients. That's always a challenge for a new modality.
Absolutely. Those are critical hurdles for any platform technology moving towards broad use.
Capstan's story really feels like it captures so many key trends in biotech right now.
I agree.
That convergence of mRNA tech spurred by the pandemic with the power of cell therapy, the massive push towards off-the-shelf scalable gene medicines instead of bespoke ones, and big pharma clearly betting big on platforms that aim for actual immune resets, maybe even cures, not just symptom control.
And it's another testament to the incredible innovation engine at places like the University of Pennsylvania, right? Where both CAR-T and fundamental mRNA tech were pioneered.
What's next? If you can give the immune system a temporary software upgrade for autoimmunity, what other complex diseases could be tackled this way? How does this change how we even think about managing chronic illness?
The possibilities feel vast. It could genuinely redefine treatment paradigms for millions.
Absolutely. So let's bring it back to that opening idea, reprogramming cells on demand. Capstan Therapeutics took that concept, which sounded futuristic just a few years ago, from a lab bench to a phase 1 trial and a multi-billion-dollar acquisition in what feels like record time.
An amazing trajectory.
Their story is just such a powerful example of how academic brilliance, focused venture funding, and strategic partnerships can accelerate breakthroughs. Now, whether CPTX-2309 ultimately becomes the first approved in vivo CAR-T for autoimmunity, well, that chapter is still being written in the clinic.
We'll be watching closely.
But regardless of that outcome, Capstan has undeniably pushed the boundaries. They've changed the conversation about what's possible in medicine and how we might treat some of our most challenging diseases. Thank you all for tuning into BioTales today. If you found this story of Capstan Therapeutics as fascinating as we did, please do rate, review, and share it with others in the biotech community. Join us next time for more stories from the cutting edge of biology, technology, and innovation.
The circuit hums with a heartbeat new. Tomorrow's edges and